today we are here to discuss the new vaccine, the conjugate pneumococcal vaccine from Serum Institute of India, and compare it as to how it compares with the existing two vaccines. So, as they say, 1 minute, yeah. As they say, all the good news come in threes. So, like these three monkeys, the first good news that came in on our way was the Covishield, the Serum Institute's and Oxford's combined vaccine was approved for emergency use on 1st of January 2021. And I'm sure, as you know, more than 10 million, that's 1 crore people have taken COVID vaccine in the country, and 90% of that has been this vaccine. So really, this has helped us to fight and save our lives. The second thing that happened was the standalone IPV, which was always there with IIP, with Serum Institute before, was relaunched. Now, this is important because this gives us now option to give an additional dose of IPV to those who have received fractional doses in the public health, or who have missed any IPV completely because they did not go to the public health, and even to use this as part of primary series or booster series in patients who cannot afford the combination vaccine. And lastly, the adults who either go to areas where they need a polio certification, or for those like stem cell transplant recipients who receive vaccines like a child, again, a standalone IPV is a boon for such fraternity. And the third and the most important good news, which is reason why we are here today to discuss, is this new pneumococcal vaccine, Pneumocil from Serum Institute of India, which was sort of dedicated to the country, not only for private use, but also for public health use by none other than our Honorable Health Minister, Dr. Harshvardhan ji, on 30th December. So three things happened back to back in vaccine field, which were very exciting. And that is why we are here to discuss the new vaccine, which gives us an option to use the vaccine in addition to the two existing vaccines. And we'll compare as to which is a best option that we have when it comes to use of conjugate pneumococcal vaccine. Before I take the science forward, the disclosures, as I always do, I'm a consultant and advisor for most of the vaccine manufacturer, including those who make either the conjugate pneumococcal vaccine or the polysaccharide pneumococcal vaccine. Now, there are certain points that I'm going to discuss as a part of the talk. The first is what is different in this new 10-valent PCV as compared to the other existing. Now, as there are two 10-valent vaccine, when I say 10V PCV, it is the Pneumocil of Serum Institute of India, and when I say PCV 10, that is the Synflorix from GSK. So that will help you to differentiate between the two vaccine. And of course, PCV 13 is unique in its own way. That's the only 13-valent vaccine from Pfizer in the name Prevnar 13. So we had this PCV7, which was launched in 2000 in USA, especially for US market, to remove the invasive pneumococcal disease in children. So the 7 common serotype, which were like causing 90% plus of the invasive pneumococcal disease in children below the age of 5 years, especially below the age of 2 years, were the target for this PCV7. And that's how you had this 7 serotype vaccine being used in the United States. And within 2 to 3 years of its use in the public health, the 7 serotype were almost wiped off from the community. And the second thing that happened was that for one child who benefited, can DIP mute everyone, please, except me? Thank you. All right. So, as I was saying, with the use of this 7-valent vaccine, there were virtual removal of all the 7 serotype causing IPD. So, the incidence of IPD dropped from say 90 to 100 per 100,000 population in children to less than last 10 or 15 cases per 100,000 population. That was like more than 90% drop. But when it had to be used all over the world, there are other serotypes which are important, which had to be taken in the vaccine, and they were especially type 1, type 5, 7F. So GSK was working on a new vaccine. It had PCV11, which was used in one what we call POET trial, where they found that the type 3 doesn't work, so they dropped it. And we'll come to this type 3 business in few slides from now. But they added this other 3 serotype, type 1, 5, and 7F, to make it 10-valent vaccine, to make it a global vaccine. And meantime, what happened in USA was that when the PCV7 was made, it was believed the 6B will cross protect against 6A and 19F will cross protect against 19A. Now that did not happen completely. While 6B did cross protect against 6A in the vaccinee, but because the cross protection doesn't induce very strong immune response, the carriage and the herd effect, which was seen with PCV7, were not seen against 6A. And for that matter, for one child who benefited because of PCV7 in general by direct vaccine benefit, there were two more cases of IPD prevented in the community, including in adults, as a part of herd effect. Now that herd effect was missing for 6A. And another serotype was emerging, which was 6C. And we knew that 6B doesn't cross protect 6C, but 6A cross protects against 6C. So 6A was added in the new vaccine by Pfizer because of direct protection, which is always better, and to have a better cross protection in adults as part of herd effect and cross protection in the form of 6C disease. And 19F did not cross protect 19A at all. In fact, when the disease burden dropped from say 90 per 100,000 to the last 8 or 9 cases per 100,000 in the United States, of this 50%, that is almost 8 per 100,000, was because of 19A. So the 19A disease went up from 2 per 100,000 before any PCV7 to 8 per 100,000. That's like fourfold higher incidence post PCV7 use. So 19A is a dreaded disease, can lead to pneumonia, parapneumonia, otitis media, sepsis, and can be drug resistant. So difficult to treat sometime. So 19A also had to be included in the vaccine. So when Pfizer made the new vaccine, they retained the 7 serotype and they added the 3 serotype, which are 157F from the global point of view. They also added type 3, which is a cause of parapneumonia, and they added 6A, 19A, as we discussed before. So it made it 13 valent vaccine. And now you have this new vaccine from Serum Institute, which is a 10 valent vaccine. So which are the 3 serotypes which are not there in this new vaccine compared to Pfizer 13 valent vaccine? It is type 4, type 18C, and type 3. And we'll discuss as to why this type 4, type 18C, and type 3 are not there. And as compared to the 10 valent vaccine of GSK, it doesn't have the type 4 and type 18C, but has the most important serotype, which are 6A and 19A. So this is a very novel vaccine which is taking care of all the serotypes which are seen globally, especially in developing part of the world, but made locally by Serum Institute of India. So we are very proud that one more vaccine has now reached its zenith, manufactured in India. As you know, Serum is the world's largest vaccine manufacturer. So why were type 4 and 18C dropped? Why was type 3 also dropped, both by the new vaccine as well as Synflorix? And why 6A and 19A are retained, and why are they important in the new vaccine? So why 4 and 18C were dropped? First of all, if you look at the Latin American countries, then you can see type 4 is hardly there, 0 or up to 1%. And if you look at 18C, again, 0 to up to 0.8%. So together, the two will be take care of around 1 to 2% of the serotype. Now, as you add more and more serotype, your immune response per serotype decreases. So say you compare PCV13 with PCV7. For the 7 common serotype, the titers are almost two-thirds in PCV13 as compared to PCV7 for the common serotype. And now Pfizer has PCV20, which is the latest vaccine which is in pipeline. And if you compare the 13 common serotype between PCV20 and PCV13, then the same story for the 13 common serotype, the titers are lower in PCV20. And if you compare PCV20 with PCV7 for the 7 common serotype, then the titers in PCV20 are almost half than that in PCV7. So you add more serotype, you reduce the immune response per serotype. So when the serotypes do not matter much, if you drop them, then you keep the number lower, which will give better immune response to the serotype which are important. So if you compare in India, we have several studies which looked at the invasive pneumococcal disease serotype distribution in the country. So you can see type 4 is 0 to 1% and type 18C again 1 to 2%. So together there'll be like 2 to 3% of the serotype, which if you drop, you don't really lose much. But by adding 6 and 19A, you make it a better vaccine. And because it takes care of a lot of serotype in the world, it's a true locally made global vaccine. Now, why type 3 was dropped by this vaccine as well as Synflorix? Now, as far as Synflorix is concerned, they did one POET trial with the PCV11, which had type 3, and they found that was a study done on otitis media, and they found that the type 3 disease actually went up after use of vaccine. So they discarded type 3 and made the current PCV10 from GSK. And same way, if you look at the type 3, which is now there only in Prevnar 13, not one but seven studies in different countries from different age group, looking at adults below 5 years, pediatric, etc., have either shown no impact or sometimes negative impact as far as type 3 disease is concerned. So except for these two small data set, one being a case control trial, which is not the best way to study, where the efficacy was there, but with a wide confidence interval, and another cohort study in Germany, where the lower confidence interval almost at zero, too. Once it becomes zero, it becomes non-significant. So both these efficacies are really not reliable. And for that, there are 8 more studies which have looked at this effectiveness, and they found that there is no impact of type 3. So type 3 really doesn't work in any vaccine. So it's a superfluous serotype in any vaccine. Now, why 6A and 19A are important? Now, as I discussed, 6A is there for direct protection for herd effect as well as cross protection against 6C, and 19F does not cross protect 19A at all. So it has to be there, 19A being an important serotype. Now, just to stress upon this, look at this new data which has recently been published in 2020 from Cireva study. That's the data on IPD in children post PCV in Latin America and Caribbean region. Now, this is looking at data of 4 countries: Brazil, Chile, Colombia, and Paraguay, which are using PCV10 of GSK, which doesn't have 6A or 19A. And you can see what happens to 19A: goes up from 35 to 131, 44 to 118, 12 to 140, and 4 to 22. So 19A disease goes up when you don't have 19A in the vaccine, like Synflorix. Now, looking at the data at other 4 countries in the same paper, which are actually using PCB13, which has 6A 19A, say Argentina, Dominican Republic, Mexico, Uruguay. Then again, if you look at this 19A, it goes down from 50 to 19. This is, of course, very small number, 79 to 45, and again 9 to 5. So while PCB10 using country, the 19A went up, whereas PCV13 using country, the 19A goes down. This shows that 19A is required in the vaccine and you cannot rely on cross protection. Now, these are the real time data from 6 countries: 3 using PCV13 in blue, Israel, USA, and UK, and 3 countries using PCV10 of GSK, Chile, New Zealand, and Finland. And you can see here, this is the direct benefit in children below the age of 5 years. Look at the blue, which are using 19A containing vaccine, and you can see the disease drastically goes down. Whereas if you look at the 3 countries using 19A missing Synflorix, then the disease goes up in New Zealand. Then they introduce PCV13 and it goes down. And Finland, it goes up and down and ultimately it goes up. So for children who are vaccinated, there is no direct effect of 19F against 19A by Synflorix, whereas Prevnar having 19A, the disease disappears. What about herd effect? So even a herd effect, you can see that 13 valent using countries, the disease above the age of 5 years caused by 19A has just disappeared from these 3 countries. Whereas you look at the Synflorix using country, consistently disease going up in these 3 countries. So 19A is required to be there in the vaccine. What about 6A? The Prevnar 13 6A containing vaccine, the disease goes down in the vaccinee. In Chile, it goes down as a herd effect, as an indirect effect, but in New Zealand, it goes up and down, and Finland again it goes down, which means there is decrease of 6A by 6B in Synflorix, and of course by 6A in the Prevnar. But you look at the herd effect above the age of 5 years, then with Prevnar, you can see the disease just disappears in older age group, whereas in Chile it goes down, but in other 2 countries it goes up. So there isn't much herd effect seen against 6A by 6B. So again, you need 6A in the vaccine, both for the direct better effect as well as for herd effect, and as I discussed, for cross protect against 6C. That's why if you look at the WHO position paper 2019, it very clearly says, as far as 19A6 are concerned, it says that setting with high 19 of 6C, you should have a 19A containing vaccine. And on type 3, it says that the only vaccine which has type 3 is Prevnar 13, and evidence from 7 countries show limited or no effect of Prevnar 13 on type 3 disease. And now you have 2 vaccines containing 6A and 19A. The Prevnar 13 are first, and also now the new vaccine from Serum Institute of being Pneumococcal. The point three to be discussed. Then what data we have? Now it's okay to have serotypes which are important, but it should also work. So what data we have for this new vaccine compared to the existing vaccine? So this is the clinical trials completed so far from phase 1 in adults, mainly to see for immediate safety. A phase 1-2 which was done in Gambia comparing with Pneumovax in adults and Prevnar 13 in children. They are using infant schedule of 3 plus 1. 6, 10, 14, 3 primary doses and a booster at 15 months, exactly what we are doing. So we will come to this 3 plus 1 schedule as to how can we use in India, and that's based on this Gambian study where it was used as 3 plus 1. Then this was phase 1-2, and based on your phase 1-2 data, you always improvise your vaccine, and you
Yenna
make
ya.
a new vaccine which then you use in your phase 3 trial. So the first phase 3 trial was done in the Gambia comparing Synflorix, where 3 plus 1 schedule was used, where 16, 14, 3 primary, but an early booster at 9 months to have better protection at the end of first year and beginning of second year, where a lot of IBD happens in children. So this was a unique study with lot to lot consistency, safety, immunogenicity, and also non-interference with the other childhood vaccination given either in the primary series or in the booster series. So based on this pivotal phase 3 trial in the Gambia, this vaccine was given international license and WHO pre-qualified. Now, the manufacturer thought that when it comes to India, they will also get Indian license based on the study. But the DCGI says that they have to do a local study using 3 plus 0 because the EPI is going to use 3 plus 0. So, and because there are two vaccines available in India, namely Prevnar and Synflorix, they had to do a small study in 3 plus 0 using comparison with both these vaccines. So, these are the new data. Now, there have been some talks by both GSK and Pfizer in recent past, where they are showing this Gambian 1-2 phase data. Now, Gambian phase 2, 1-2 data is not to be included or not to be seen actually because after this, the vaccine has been tweaked or vaccine has been improvised. So, what you should see is actually the phase 3 data because what vaccine we have is what was used in phase 3 and not this phase 1-2. And this phase 3 data from Gambia is recently published. So, all of you can actually see. And phase 3 Indian is not published. So, it's data on file. So, I have the privy to have this data to see that, and I'm going to show you. So, you are like seeing data which is not even published. So don't go by phase 1-2 of Gambia where the vaccines look different as compared to phase 3. Phase 3 is the most important. So I'm going to show you both this phase 3, the published data from Gambia and the unpublished data on file from the Indian phase 3. So how was the new license vaccine, this new vaccine license globally and WHO qualified? So we are going to first look at the international licensing, and that was based on the Gambian study phase 3. And before we dissect the data, two things are important. As far as the regulators are concerned for licensing, we know that the new vaccines cannot undergo efficacy trial, how it happened with Prevnar 7. Even a Prevnar 13 did not have efficacy trial when it was launched. Because one, it is unethical to do a placebo controlled trial because we have life-saving vaccines available. Two, the countries which can do this type of trial, the Western country, they will not allow any efficacy data further. So WHO came to the rescue for the new manufacturer, including for Prevnar 13. They don't have to do this efficacy trial, which are so expensive and so laborious, but they decided a cutoff of IgG response at 0.35 microgram per ml as this criterion for non-inferiority on immune response compared with the existing vaccine. So that's how these vaccines were licensed. But when you look at IgG, those may be actually functional or they may be non-functional. Most of them are functional, but sometimes they are not functional. So how do you know antibody is functional or no is what we are interested as clinician, that whether this antibody is going to kill a organism in vivo when you inject the vaccine in the body and it makes antibody. So that is what we call functional assay or opsonophagocytic assay or OPA. What do you do in OPA? You take the serum of the vaccinated person, which has say antibody against, let's say 19A. Then you take a 19A live pneumococcus and in vitro. You add the serum and you add human complement. Now, with this antibody against 19A is functional, it will kill that 19A organism. So at what dilution it kills 50% will decide the titer or GMT. Now, why am I stressing so much about IgG versus OPA? IgG is the requirement of a regulator for licensing. OPA is the requirement of clinicians like us for that antibody to be working or functional. Just to give you an example, look at 19A. The red are PCV7 data and the blue is PCV13. PCV13 has 19A, so obviously it's going to work for 19A. But you look at the IgG response. Sorry, this has to go 1 minute. Yeah, okay. So the blue in PCV13 has 19A, so almost nearly 98% seroconversion against 19A. You can see nearly 100%. But look at PCB7, which doesn't have 19A, has a lot of IgG antibody in 80% of the subject as a cross reaction between 19F and 19A. But and that is in the German study. There was one more study in USA where similar study, almost 98% with PCB13 and 85% with PCB7 when PCB7 doesn't have 19A. Now, if there is so much of robust immune response against 19A by PCB7, then why did it not work? As we discussed, the 19A disease went out of the hand. In fact, it increased 4 times and it became 50% of the remaining disease burden is because these antibodies were not functional. So how do you know? So this is looking at the post-primary OPAS. You can look at these 19A, PCV7, hardly any titer. Look at PCV13, huge titers. Again, you look here, 19, hardly any titer, PCV13, huge titers. So what is important is that these antibodies have to be functional. So based on that, we will look at this data on the phase 3 trial, which led to international licensing. So a phase 3 trial, double-blind randomized control trial, comparing along with against Synflorix for a lot of things, lot-to-lot consistency, immune response, safety, and non-interference with the other concomitant vaccination. So looking at the safety profile, you can see the local side effect of induration and swelling was 4 to 5% with Pneumococcal and almost 10% with Synflorix. So half with Pneumococcal compared to Synflorix. All other local side effects or the systemic side effect, there was no difference. So there was less swelling with Pneumococcal as compared to Synflorix is the conclusion. Looking at the IgG response post-primary, this is the difference in the seroconversion cutoff being 0.35, and this is the ratio of Pneumococcal versus Synflorix on the IgG titers. So say for example, your seroconversion with type 5 was 98.4% with Pneumococcal, it was 95.6% with Synflorix, the difference being 2.9. So all which are shown in black, which are more than 0, are numerically better. And there are 7 serotypes which are numerically better. But if the lower confidence interval is touching 0 like here, that is not significant. But like here, it's above 0, that is significant. So 4 serotype, type 5, type 23A, 6A, 19A was significantly better seroconversion with Pneumococcal compared to Synflorix. Now, if you look at the titer ratio, so say for type 1, the ratio is, the titer is 4.29 with Pneumococcal, a 1.9 with with Synflorix. So it's almost like more than double. So you can see the ratio is 2.115. Now again, the confidence intervals, if this touches 0, this value is not significant, though numerically better. But if it is above 0, it is even statistically significant. So like see here, the ratio is more than 1. But it's not significant because the lower bound confidence interval is touching less than 1. So if you look numerically, the ratio was better for 7 to 8 serotype and statistically significantly better for 7 of these 10 serotype. So a lot of serotypes showing better seroconversion and definitely better GMC ratio with Pneumococcal compared to Synflorix. Now the two additional serotypes, 6A and 19A, look at the difference. 73% more with Pneumococcal compared to Synflorix and 55% more with Pneumococcal compared to Synflorix for 6A and 19A respectively. And the titer ratio, 8 times more with 6A and 6 times more with 19A. So no doubt these two serotypes being there in the vaccine are very powerfully immunogenic. Now what about the OPA? Because to us OPA is more important. So first of all, two serotypes, type 1 and type 5. The OPA seroconversion was less than 80% with Synflorix. These are two important serotypes. Whereas for Pneumococcal, none of the 10 serotypes were less than 90%. And if you look at the difference in the OPA seroconversion, 4 serotype were statistically significantly better, namely 1, 6B, 6A, and 19A. And if you look at the titer ratio, then for 2 serotype, 9V and 19F, the titers ratio were better with synflorix compared to pneumocil. But for 7 serotype, they were statistically significantly better with pneumocil compared to synflorix. And now we're going to follow up this 9V and 19F serotype, which seem to be a little better with synflorix compared to pneumocil. What happens to them post booster? So this is the post booster response. You can see here, first of all, for some reason, type 5 did not boost with both the vaccine. As you can see, the ratio was, you see the post booster ratio was 1.31 for pneumocil, which was 1.49 post primary. So, of course, did not boost. And for synflorix, 1.26 became 0.84. But yet, the ratio between the two vaccines was in favor of pneumocil. And like that, again, for the two serotype, 9V and 19F on IgG response seem to be better with synflorix. For all other 8 serotype, they were better with pneumocil compared to synflorix. And these two serotype, what happens to the functional antibody, the OPA? So you can, these are the OPA titers. Those two differences disappear. And for the remaining 8 serotype, statistically significantly better with pneumocil compared to synflorix. So the end of the booster response, not a single serotype is lesser with pneumocil, whereas 8 serotype are better with pneumocil compared to synflorix. And that led to WHO pre-qualifying this vaccine way back in December 2019, and this vaccine getting international license as 3+1 schedule for use in several countries through WHO and UNICEF. Then how was the vaccine license in India? As we said, the DCGI did not give permission based on this Gambian phase 3 trial or the international licensing trial. So they had to do a quick study comparing this vaccine with both the existing vaccine, namely Prevnar and Synflorix, and 3+0 schedule, because that's the EPI schedule which is used in India. 8 different sites in India were part of this study. So if you look at the side effect profile, local side effect, if you compare pneumocil with Prevnar, there was no difference. Whereas you compare pneumocil with Synflorix, then local tenderness was again half in pneumocil compared to Synflorix. So in the Gambian phase 3 data, local induration was half, and in Indian phase 3 data, tenderness is half. So local side effects seem to be lesser in pneumocil compared to Synflorix, but as compared to Prevnar, they were the same. And for the systemic side effect, whether you compare pneumocil with Prevnar or pneumocil with Synflorix, there was no significant difference. So the vaccine was as safe. Now coming to the seroconversion and OPA, first set of slide will be comparing it with the Prevnar 13, and the next set of slide will be comparing with Synflorix, as two vaccines were a comparator. And this being 3+0, all are post-primary, as there was no booster given in this study. So this is post-primary IgG seroconversion above 0.35. Comparing to Prevnar, you can see two serotype were better with Prevnar on IgG response. But when it comes to OPA response, that difference disappeared. So on OPA, a functional antibody response compared to Prevnar 13, post-primary seroconversion was just the same between the two, if at all numerically better for at least 5 serotype in pneumocil compared to Prevnar. What about, sorry, just 1 minute. Yeah. What about the post-primary? Just 1 minute. Yeah. So this was a post-primary IgG and OPA seroconversion, as I said, compared to Prevnar 13, where there was no difference between the two vaccines, numerically better for 5 serotype compared to Prevnar 13. Now, what about the post-primary GMC ratios and the OPA ratios? For 7 serotype, the GMC IgG ratios were better in Prevnar compared to pneumocil. But when it comes to functional antibody, OPA ratios, for all the serotype, the difference disappeared. In fact, for most of them, it was numerically better in pneumocil compared to Prevnar 13. And for one of them, it was statistically significantly better with pneumocil compared to Prevnar. So on OPA GMT, whether it is seroconversion, there was no difference. And on OPA titer ratio, numerically better for most of the serotype and statistically significantly better for one of the serotype. Now compare with the Synflorix, post-primary IgG seroconversion, better for 4 serotype. For OPA, one better with Synflorix, one better for Pneumococcal, so nothing much difference. When it comes to IgG ratios, for one serotype 19F, it is better with Synflorix, but that difference disappears on OPA ratios, whereas for 7 serotype on IgG response and 2 serotype on OPA response, significantly better with Pneumococcal compared to Synflorix, and most of the serotype were numerically better in Pneumococcal compared to Synflorix. So again, like what happened in Gambian study, a lot of serotypes showing good response or better response with Pneumococcal compared to uh, Synflorix. Now, burning question, based on all this data, can you use this vaccine, new vaccine 3+1, where Indian study was 3+0? So phase 1 to Gambian data uses the 3+1 with 3 primary at 16, 14 and the booster at 15 months, exactly what we are using. Global licensing is for 3+1. WHO has pre-qualified for all the schedule, including 3+1. And we have an Indian toddler study comparing new vaccine with the Prevnar 13 for naive children who have not received any pneumococcal vaccine in the past, who come in the second year. What happens when you give these two vaccines and compare, giving two doses 8 weeks apart? So that's what I'm going to show you, the phase 2 toddler study done in India at 3 sites where children who are naive, who had not received any pneumococcal vaccine, at 12 to 15 months received either Pneumococcal or Prevnar 13, 2 doses 8 weeks apart. Now, if you look at the data, sorry, my computer is hanging for some reason. We'll have to wait for some time. 1 minute. I hope I don't have to restart it. Just give some time. Yeah, 1 minute. Sorry. What happened? Yeah, 1 minute. I'll restart sharing the screen. Something happened in the, I think, 1 minute. Just give me 1 second. Now I do it again. Are you able to see it now?
Not able to see,
No,
sir.
doctor.
Yes, you will see very soon. 1 minute.
Yes,
Fine.
it's visible now.
Yeah. So this is where I was. I want to show you the toddler study. So naive children, 12 to 15 months, received 2 doses, 8 weeks apart. So this is the safety data. Post dose one, post dose two are combined. There was absolutely no difference between the two vaccine as far as any side effect profile is concerned. Coming to the IgG seroconversion and OPA seroconversion, one serotype was better with Prevnar 13 on IgG response, but that difference disappeared when it comes to OPA response. So on OPA response, there was no difference between Pneumococcal or Prevnar 13 in these toddlers who are naive. What about the ratios of the GMC, IgG, and OPA? On IgG, GMC ratio, 5 serotype were better with Prevnar 13 compared to Pneumococcal. But when it comes to the OPA functional antibody, except one 6A, all other four serotype, the difference disappeared. And in fact, for some of them, it was numerically better. So the conclusion of the study was that at the end of the second dose, on functional antibody response, there was no difference between the two vaccines. They worked equally well. That is the reason why, if you see the latest ACVIP guideline 2020, very clearly says that it takes cognition of pneumococcal and it approves the use of pneumococcal to 2 years of age in 3+1 schedule with the booster given at 12 to 18 months. So there is absolutely no reason why you should get worried medico-legally or otherwise about using the booster dose. If you give an extra dose, there is never a problem. If you give less dose, there is a problem. So you have WHO backing, you have the Gambian phase 1-2 backing, you have the Gambian phase 3 backing, you have the ACVIP backing to say that you can use and you should use this vaccine as 3+1. Now, the last few points: how will the new vaccine compare on the serotype coverage with the existing vaccine? So, you see, globally, around 71% of the serotype are covered by this vaccine, and India nearly 73%. And if you look at the Indian data, then we have not one but four different data sets. So, except PDOPs, where they all three look similar, for all other data, you can see here the 6A 19A containing vaccine, namely Prevnar 13 in blue and in pneumococcal in red, as compared to Synflorix, which doesn't have 6A 19A. See the difference: 70 to 71 very similar between 6A 19A containing vaccine, 8% less with Synflorix. Same way here, 78, 77, and 64. Here 76.6, 73, and 67. So almost 7 to 8% lesser with non 6A 19A containing vaccine, and between the two vaccine with 6A 19A, there isn't much difference. 1% here, 1% here, some 2% here. Very recent data done in Haryana, Palwal region, where they took children who were healthy or children who had community acquired pneumonia, and they took their nasopharyngeal swab to see how much of these three vaccines will cover for the serotype. So for healthy children, again, there was not much difference between the 6A and 19A containing vaccine. It was 2% less with Synflorix. But when it comes to diseased children, you can see here it's 32 with Prevnar, 31 with pneumococcal, and straight 24%, 7 to 8% lesser with Synflorix. So that difference will always remain. So what are the future studies planned? So as India is using 6 weeks, 14 weeks, and 9 months, and a lot of international countries are going to use same 6, 14, and 9 months as early booster schedule. So two studies are planned: phase 3 in India using 2+1, comparing with Synflorix and Prevnar, and a Gambian study again 2+1 comparing with Prevnar and Synflorix. This data, because of COVID, is delayed. I'm sure it will come in next few months. A post-licensure impact study in India, Indonesia, and Vietnam, where children will be studied, 1,200 children, for their carriage to see for serotype before and after vaccination to see the impact. And any rare side effect will be picked up in this pharmacovigilance, or what you call phase 4 trial, which is a mandatory trial for any new vaccine. And this is the opinion by Dr. Cynthia Whitney. For those of you who don't know, she was at the helms of the affair in US CDC when Prevnar and then Prevnar 13 was launched, right from 2000. No wonder she has more than 230 publications on pneumococcal disease alone. Now she retired from there and joined Emory University, and this is what she has to say. Across studies, this new vaccine meets all immunogenicity and safety data following primary and booster dose. It has a very safe reactogenicity and a good immune response similar to the licensed vaccine. It has shown lot-to-lot consistency and non-interference with other childhood vaccination when given simultaneously. And overall, because of this data, it says that new vaccine performance, if incorporated in the national program, will be similar to that of the existing two vaccines, which have had major impact on pneumococcal disease. So the question that comes, there's a new vaccine, we don't have data from any country on effectiveness. All new vaccines which come don't have data on the effectiveness. Like Prevnar 13 didn't have, Synflorix didn't have. They all had over last 10 years of use. So the same thing, we will accumulate data on this new vaccine once it's being used in the national program in several parts of the country. So I will just end with these two mission statement by our Honorable Prime Minister Narendra Modi ji. In his first tenure, he said, "Make in India," and in the second tenure, he is saying, "Atmanirbhar Bharat." So we make as much as possible in our country, and I'm happy to say that this is Make in India Atmanirbhar vaccine, which has all the global serotype and a vaccine done made globally by Serum Institute of India. So be Indian, buy Indian, Atmanirbhar. If you believe that, one more reason to use this vaccine, besides the cost consideration, which of course in a scientific talk we don't discuss, but we can discuss in the question answers. Well, thank you very much for patient hearing. I'll stop sharing my,
Thank you, sir.
we can take question answers.
Yeah. And thank you, sir. Dr. Nitin, sir. I think you nicely compared the Pneumococcal with the Synflorix and as well as PCV13 also. I think you have elaborated post-primary response better in Pneumococcal as well as the local side effects also less in the Pneumococcal and better IG response. And another thing is coverage by same compared to PCV13 and PCV10 also, Synflorix. And this Pneumococcal is definitely it's a better immunogenicity and safety. More than that, it may be incorporated into the national immunization program cost-wise also. Definitely, the cost-wise is better, and it is definitely it's a wonderful your input to our pediatric community. Thank you, sir. Hey, anybody, any questions, please? Audience side. Dr. Vendesh, sir, Dr. Sudhakar, any questions?
What is
So
the
there is a
cost?
new one question audience. What will be the cost of new vaccine, serum vaccine?
Correct. So the Prevnar, as we know, Prevnar 13 is around 4,000 plus MRP and around 3,300 or something doctor's price. Synflorix has recently jacked up its price, I think, to some 2,000 rupees, and doctor's price is some 1,400 or so. And Pneumococcal, the MRP is 1990, I think, and the cost to the doctor is around 1,100. So it's almost to 300 lesser than Synflorix and almost less than half or less than one third of the cost of Prevnar 13. And this vaccine is going to be part of the national program because, as I understand, in the current budget, 35,000 crores have been set aside for pneumococcal conjugate vaccine, and I think 25 million doses order from the government has been placed with Serum Institute of India to supply pneumococcal. So very soon, Prevnar 13, which is part of the national program in some states, 5 states, will also be replaced by pneumococcal, and more states will be incorporated. So the idea is in the next 1 or 2 years, all over the country, you will have conjugate pneumococcal vaccine available through national program.
I think we can grassroots level, that's very important. The basic level we can take to a rural area, it's easy to administer as well as cost wise also definitely is much cheaper.
Absolutely. So see, so far we had Prevnar 13 and Synflorix. Now, Prevnar 13 is 6A, 19A, and a lot of data from all over the world. So we would want to use a 6A, 19A containing vaccine for all the reasons that I showed you. But the cost was so high that for many patients we could not use. So we were using Synflorix even when it doesn't have 6A, 19A. But now when you have a vaccine which is 6A, 19A, and is also even cheaper than most all the other two brands, I mean, it makes complete sense. And it being Indian and Atmanirbhar, if you believe in all those nationality, patriotic type of feeling besides science. So on science, it is equal to Prevnar 13. On cost, it is one third, and on national pride, it is an Indian vaccine. It makes a perfect match or combination, I would say, to say that we should use this vaccine. So those who want to take Prevnar 13, they can continue to take. But now, when you have 6 and 19 containing vaccine, to give Synflorix, which doesn't have 6 and 19, really doesn't make sense according to me.
Sudhakar, any questions?
One question from the delegates. Is this can be given for adult actually, just like in Prevenar in the old age people and immunocompromised? Is it going to be useful for the adult also?
Correct. So this vaccine has study only till 2 years of age and not beyond. So we cannot use this vaccine for even children above the age of 2 years as catch-up. Whereas you can use Synflorix and Prevnar for 2 to 5-year-old children. And Prevnar is like womb to tomb. You can use right from birth till adulthood. So this vaccine is going to have further studies on 2 to 5-year-old children, immune compromised children, and in adults. So till the time that data accumulates, which will take few years, we should not use this vaccine for beyond 2 years of age.
So in the current, another question, the current scenario, in Tamil Nadu, this Prevenar has been, pneumococcal 13 has been given for the newborn babies less than 1.5 kg.
Correct.
What will be the strategy when they introduce pneumococcal to the government scheduled.
So you mean to say in Tamil Nadu, it's only given for those who are below 1.5. For above 1.5, 2 kilos.
As of now, it has been given only in that base.
Acha. So what it means is that it is not become part of national program in Tamil Nadu. As you know, the national program for PCV has been started in states where there is more childhood mortality. That's how it went into northern and eastern states more. But sooner or later it will come to western and southern states also. Now, just because there is a lot of mortality and morbidity in babies who are born small, as a pilot, they've started giving the targeted pneumococcal vaccine for those age group. But once now in this budget, as the money has been given for inclusion of this vaccine in the national program all over the world country, I think sooner or later all Prevnar 13 is going to be replaced by Pneumococcal. So even it will happen for even those small babies.
Okay, sir. Thank you, sir. Now I'd invite, sir, one question. Of course, we may not be able to answer this question. If somebody has started with Prevenar, can we? That's exchanging the vaccination.
So normally we say that by choice you don't change the brand, but by default you can. Now in question coming of, suppose someone has taken 1 dose of Prevnar and he says, I've heard that there is a vaccine which is very cheap and as good. Can you, what do you do? If the parents insist, certainly give them the other doses of Pneumococcal. There is no problem, but not by choice. But do not give from 6A 19A containing vaccine to a non 6A 19A. So I wouldn't change from Pneumococcal to Synflorix or Prevnar to Synflorix. But I can change from Synflorix to Pneumococcal and Prevnar to Pneumococcal if the parents insist because of the cost consideration.
Yeah.
Okay. That's all questions are over, sir.
All right. Thank you very much.
On behalf of IAP TNSC, I thank Dr. Nitin Shah sir and has spared our valuable time. And after a long time, we are seeing Tamil Nadu, and I really thankful to you, sir. And I thank Dr. Vendesh sir, Dr. Sagar, and Gopal Subramaniam, and Dr. Karamat, and other seniors who have given this opportunity. Thank you, one another. Thank you.
Thank you, everybody.
Thank you.
Thank you, Nitin.
Bye bye. Bye bye.
Bye bye.